Great. Hi, everybody. Can you hear me? Okay. Hello, welcome. Thank you for waiting. I am presenting Angie Lenard here today. Angie is coming to us from the US. A little bit of background on Angie. Angie did their master's at University of Central Arkansas, working on Scaloporus lizards and embryonic development. They went on to work on butterflies and their thermal biology in the context of urbanization at Case Western Reserve University, which is where I know Angie. Angie and I did our PhD together in the Department of Biology at Case Western. They are now a postdoc at the University of Nevada, Reno, working with Matt Forrester. Currently, they're conducting butterfly monitoring and pesticide discovery in national wildlife refuges. So that's what they're going to be talking about today. So it's going to be an exciting talk. Everyone, welcome Angie.
Thank you so much. Is my microphone on? Okay, perfect. I can never tell. Because my own voice is so loud in my own head. So I'm glad that hopefully everyone online can hear me too. So I'm really, really excited to be here today to share some of the pieces of the mosaic that make up my research program. And so I'm interested in human-modified environments because they expose organisms to novel stressors or novel combinations of stressors. And we're still working really to understand the exact challenges that organisms are facing in these environments and how they might be overcoming them. And so I use insects as a focal model system in my work to document challenges that organisms face in these human-modified environments, such as cities and agricultural regions. And I've used the cabbage white butterfly also as a focal system in order to ask how traits could be changing through contemporary evolution or phenotypic plasticity to help this species overcome these types of challenges. And so I apologize if any of the graphics, the transfer from my computer to this one made things maybe a little blurry or different fonts. So hopefully there's nothing too weird in the talk. But we know that insects are facing a multitude of threats to their biodiversity. So ranging from habitat loss to climatic shifts to alterations to biotic interactions. And while each of these threats undoubtedly is contributing to the insect biodiversity crisis, the actual picture is much more complex. And so this figure is a meta-analysis of meta-analyses, and it shows how often terms are discussed in the literature of insect declines. And so longer arcs around the circle show terms that are discussed more often, and then you can trace the connections between these terms to see their relative contributions and the underlying causes. So for example, if you're looking at land use change in green on the bottom, about a third of that is explained by agriculture if you follow that purple section back to the agricultural arc. And so changes to habitat quality and to land use are often dominating the discussion, but a lot of times these terms are used as intermediaries to discuss things like urbanization and agricultural intensification. So in my research program, I focus on these human-modified environments because they're linked to multiple of these important stressors. And I also believe that in order to understand the insect decline, we need to study these environments because they're going to be essential for balancing societal needs with insect conservation. And Lepidoptera declines are especially well documented, so are moths and butterflies. And while they are not experiencing the most severe declines compared to all invertebrates, we simply have the most data on this group, so we can pull out some specific trends for Lepidoptera. And one of these patterns reveals that butterflies that are in disturbed areas tend to have lower diversity communities. And the reason that we know so much about butterflies is because of monitoring efforts. So there are programs across the globe where folks go out and count butterflies and watch their populations over time. I'm just highlighting some of the work that I do with my collaborators in the Western United States at the University of Nevada, Reno, where we've been running long-term monitoring programs for butterflies and caterpillars. And then we also do participate in national counts like the NABA, North American Butterfly Association counts that do annual 4th of July. monitoring programs, and also taking community members out through Earthwatch programs. And so with this wealth of data, we are able to understand butterfly declines a little bit better, and the drivers that are causing them. So for example, this is some work from my collaborators where we have been using this long-term data set from the Western United States that has monitored butterflies in California for the past 50 years. This was started by the University of California, Davis professor Art Shapiro back in the 1970s. And so I'm showing you just some data from 5 of those sites in the California Central Valley. And these data agree with the fact that monitoring has revealed that there are these long-term declines in populations. And what I do and my collaborators do is we work to understand what's driving those declines. And so this data is very valuable in being able to piece apart the relative effects of different anthropogenic stressors. And so, for example, if we zoom in on this steep portion of the decline prior to 2005, and go back in time a little bit more to the beginning of this monitoring effort in the 1970s, we see that this analysis reveals a kind of break point where the decline really started in 1997, which is just 2 years after the introduction of neonicotinoid insecticides on the landscape. And so pesticides and land conversion are both really important drivers of butterfly decline. So when we piece them out of this data set, we see that they have the largest negative coefficients explaining the variation in butterfly diversity in comparison to things like minimum temperature and sampling effort, which overlap with 0. And both of those variables are important in agricultural and urban environments, but we still have a lot of work to do to understand them in these spaces. So the fields of urban ecology and urban evolution are fairly young and new, and so we're still documenting contemporary evolved responses. And then in terms of pesticides, the impact of pesticide contamination on green spaces such as parks or natural wildlife refuges or these large areas that could be used as butterfly habitat are relatively unknown. And then even when we do know what pesticides are in those spaces, very often we don't actually know how they affect non-target pollinators. And so, oh, all of that disappeared. That's okay. So today I'm going to tell you 2 short stories. Hopefully it's cut off a little. But first, I'm going to focus on what stressors are pollinators facing across these gradients of disturbance. So as we move from a natural space into more of a modified environment, what kind of stress are they experiencing? This work will take place in the California Central Valley, and so I will be talking about how I've documented pesticide contamination in both agricultural and urban landscapes. And then next I'll move into responses. So how do pollinators actually respond across these disturbance gradients in terms of the evolution or plasticity of their traits? And in this I'll focus on work that I've done with urbanization in the state of Ohio. And this will be a multi-trait examination as well as looking at the relationships between these traits. All right, so our first story brings us to the Sacramento National Wildlife Refuge complex. And so this is a refuge complex in the northern part of the state of California. And so prior work in this region revealed that milkweed plants showed ubiquitous contamination. So milkweed is the host plant for the monarch butterfly, which is of conservation concern. And basically when we went out and tested for contamination in like the edges of wildlife refuges, agricultural regions, urban regions, or natural spaces, we found that every single plant was contaminated with some kind of pesticide that could be detrimental to monarch caterpillars. So it really raised the alarms, and this paper got a lot of attention because it made people wonder if there are any good protected areas for butterflies in this heavy agricultural region. And so I partnered with the United States Fish and Wildlife Service to study their lands. So they have wildlife refuges across the Central Valley in this heavy agricultural area. And they are these large parcels of land that they set aside mostly for bird conservation, but the state has really been getting very interested in conserving insects as well. So we worked together to understand what the insect populations looked like on their landscapes, but also to see how much pesticide was making it into the landscape. And this is an especially important question for these protected areas because they are just heavily surrounded by agriculture. So these are 5 properties that I worked at, and here I'm showing you some of the surrounding agriculture. So the refuge lands are shown in gray, and then the 2 dominant crops in the area are rice and orchards to make almonds and walnuts mostly. And so you can see that there's a lot of agricultural activity. The rice is shown in yellow, the orchards are in purple, and right next to these refuges there's a lot of agriculture. So we went across the refuge landscape and put out these pesticide plots where we were testing for the occurrence and the concentration of different pesticides, both at the refuge edges where we're very close to agricultural activity, as well as the refuge interiors. And I want to thank my collaborators at the United States Geological Survey, Wayne Thogmarin and Michelle Hladek. They were providing resources such as these passive samplers. And so on the end of this orange pin flag, you'll see it's a silicone wrist bracelet. And so it's kind of plant colored, so it's the green, black, and yellow thing that the arrow is pointing to. But this is a very cool technology where it's very simple. It's just like the silicone wrist bracelets that people use for activism. And you can put them out into the field clean of pesticides, let it sit out for a period of time, and pesticides will accumulate on the surface as they drift through the air. And then you can pick them up and wash off all of the pesticides and see what's on the landscape. And so it's a nice way to get like a metabolically inert snapshot of a period of time. And so we put these out early in the season at all of our pesticide plots. And I love showing this picture because it shows this is one of the edge plots where we got really close to agriculture. So in the foreground of the picture, you can see the flags where the pesticide bans are hanging. And then in the background, you can see them preparing a rice field for planting. So like right across the sidewalk, essentially. It was a dirt road, but right across the dirt road, we had them preparing an agricultural activity. But to contrast that, we also had these really nice interior landscapes where we were testing for pesticides as well, and lots of floral resources for the pollinators, and even some of these really beautiful vernal pool habitats that had some endemic California calico flowers. So before I show you everything that we found on the bands, I want to walk you through this graph. So on the left-hand y-axis, you have the names of all of the different pesticide compounds that we discovered. So we screened for way more than this. We screened for almost 200 different compounds, and we found 36 different compounds that popped up at some site. And then on the right-hand y-axis, we see whether that compound was an insecticide, herbicide, or a fungicide. And then I'll populate a bunch of different columns. And so in each column will show a separate plot. And then you can see on the x-axis whether that plot was on the edge of the refuge or in the interior. And then the 3-digit codes at the bottom are just the different refuges. The colors that show up will show the amount of nanograms of pesticide that we found at that plot, and the darker coloration indicates more pesticide. So this is everything that we found, and I want you to take home 2 major messages. The first is that this is a lot of pesticide compounds within each plot. So across our columns, the average number of compounds that we found was 18 different pesticide products. The maximum, the most contaminated plot had 24, and then at a minimum, so our cleanest plot had 12 different pesticide products. And these were combinations of insecticides, herbicides, and fungicides, so pollinators navigating these habitats are likely exposed to mixtures of a lot of different products. The other thing that I want you to take away from this is that we didn't see any real differences between the edge and the interiors of these refuges. And so on the edge, we see the same compounds popping up in the interior. And there's not a significant lightening of the coloration in any of our refuges, which shows that there's roughly the same amount of pesticide making it into the interior plots as on the edge. But this is a very kind of coarse categorization just into edge and interior. and not every single plot was right next to a rice field like in that picture I showed you earlier. So I went in, I actually calculated the distance from each of those plots to the nearest agricultural activity and found a very similar message. And so the distance from the agricultural activity was largely unimportant in explaining the pesticide presence and the amount. So I'm just showing you here the number of unique pesticides, or what I'm calling pesticide richness, but this same pattern held if we looked at the total nanograms of pesticide present as well. And so I've broken this out into their different pesticide classes, so fungicides, herbicides, insecticides, and we had one synergist, which is just a compound that gets added to increase the efficacy of insecticides at killing insects. And you can see that most of these slopes are not significantly different from zero, and so there really was no effect of distance. And this was especially true for when we had the closest crop type being rice. And so the rice, I've noted at the top of these whether rice or orchard was our closest agricultural activity. And so the only difference we saw was that in the orchards, there was a decrease in the number of fungicides if you moved away from the refuge edges. And this is nothing special about national wildlife refuges or these protected lands. They're in a very heavy agricultural region, so even though we were able to do the work on these lands, this would be a problem to any lands in this area. So including the cities in the regions where all of the workers are living, as well as, you know, even some of the major cities. So we repeated some similar work. I don't have time to talk about this project at length today, but I just wanted to note that this is not a problem with the wildlife refuges themselves. So we went to the city of Sacramento, California, which is just to the southeast a bit of where our refuges are, and we sampled a bunch of different plant genera in order to test the host plants for the pesticide contamination as well. And this was a collaboration done with PhD student Claire Ditmore, as well as the Xerces Society for Invertebrate Conservation. And we found a very similar thing where all of the plants had levels of pesticide contamination, a mixture of insecticides, fungicides, and herbicides that were at concerning levels for caterpillars of different butterfly species. And so I'm really excited to get this data back soon, because in addition to using the silicone bands, for the past 3 years, I've also been going out to sample host plant tissues at these same wildlife refuge properties. And so there's a lot of data that went into this. And host plants will be really good because they provide a different perspective. So while these silicone bands are a nice look at the aerial deposition of pesticides, host plants are also going to be picking up compounds from the soil or water sources, as well as the plants themselves can metabolize the compounds, and in that case kind of mediate what the caterpillars are actually exposed to when they're eating those plants. So this study is kind of unusual in the literature based on the number of host plant species. I was able to completely capture the diversity of all host plants, that every single species of butterfly that we know uses these wildlife refuges for habitat. And so it's going to be one of the most holistic and ecologically inspired studies of its kind. And so I'm very excited. This data should be in my hands this fall, so look out for updates on that. All right. So that brings me to the end of our first story, kind of a surprising find in that we found that there was no real gradient of contamination across this space, that they're likely to be experiencing these anthropogenic stressors even in the interiors of these natural protected lands. So now I'm going to switch gears a little bit and talk about how are pollinators responding to disturbance gradients, and specifically in terms of urbanization. And I am going to focus on the Pyrus rapi, which is the cabbage white butterfly, because it's really excellent at persisting in modified environments. And so this is data, I mentioned I was also looking at the butterflies on these wildlife refuges. So this is data showing for the past 3 summers the abundance of this butterfly at 8 different properties that I managed, or that I monitored. And so in this case, it's found across all of these. And so these are a wide range of habitats. They're all, pesticide covered based on our previous results. And so this butterfly does really well at living in these agricultural landscapes. It's an agricultural pest of brassicas. And so it's persisting quite well in the agricultural sphere, but also in the urban sphere. So I don't expect you to be able to read all of that text on the left-hand side, but those are all different butterfly species names. And then on the x-axis is the urban affinity score that was assigned to them based on their likelihood to hang out in cities in Europe. And so these researchers went and actually looked to see which ones are avoiding urban areas, which have a negative urban affinity score, and that's most butterflies. But some butterflies actually preferentially hang out in urban spaces, and at the very top is Pyrus rapi, our cabbage white butterfly. And while this is anecdotal, this just feels right to me. They are, sorry, I can hear the buzzing a little bit. I hope that's not too bad. They are this very common, urban species. I used to see them in my garden in Cleveland, Ohio. And I would see them overwintering on the siding of my garage. So they were just really amazing at living and using these like human modified spaces. Oh, I can see the next slide looks weird. Okay, none of the clips transferred, but you can maybe imagine. Urban organisms are facing various stressors when they're living in urban environments. So I already mentioned that in a city we found similar pollution of pesticides, but there's also other contaminants, right, from like motor vehicles and putting smog into the air and factories. So they're dealing with pollution. They're also dealing with artificial light at night. So if you are interested in that, you should definitely talk to Troy, who introduced me. They're doing an artificial light at night experiment. Do you want to just switch over?
After
Okay. Thank you. This is on? Okay. And then they have dealing with the urban heat island effect. So the instance where urban spaces are warmer than the natural and surrounding environment. There's altered biotic communities. So based on the plants that people are choosing to plant right in the cities or the pests that they're choosing to control for, we're going to get altered biotic communities. And then finally also habitat fragmentation. So that's a
know just by looking at them in their respective environments if that was from evolution. We would have to take them, we're going to go through some animations. We're going to take them out of those environments, rear them under common conditions, and then if we still see that those phenotypic differences are persisting, we could then infer that it's from evolutionary change. And so that's exactly what I did using common garden experiments across the city of Cleveland. So this is a map of Ohio in the Midwestern United States, and we're up in that small box on the coast. So this is, the white is actually water, that's Lake Erie, one of our Great Lakes. And then I went across an urbanization gradient where the more urban spaces are shown in dark red, and then the more rural spaces are shown in the grayish blue colors. And so I collected female butterflies from across this urbanization gradient. I allowed them to lay eggs on host plant in the lab, and then I separated those eggs in a split clutch common garden design into two different common garden temperatures, a cool temperature and a warm temperature. And this design, what it allows me to do is to look for plasticity by looking within a population. I can see how the temperature treatment is affecting the development of those phenotypes. I can also look for evolution. So by looking within a temperature treatment, how do the different females from different populations produce different phenotypes? So are they differing in the same common garden temperature? And then I can also look for the evolution of plasticity itself. So in that case, you can look to see whether the reaction norm or the difference right between the cool and warm temperatures for any given population is different. And so that's a lot to break down, I know. So I'm going to walk through kind of graphically how this will show up in all of my results slides. And so in this case, if we're looking at a phenotype that's plastic, we have our two different common garden rearing temperatures on the x-axis. So we raise them in cool and warm conditions. And you can see that some phenotype of interest is plastic. So they're developing different phenotypes in those different common gardens. But the rural and urban butterflies are always showing the same phenotype, right? So in this case, we're not getting any effect of evolution, and it's just a plastic trait. We could also have the situation of evolution only. And so in this example, the phenotypes are fixed. So the rural like circles are always presenting the same phenotype regardless of the developmental temperature. And then the rural, urban ones are doing the same thing. But you can see that there's a really big difference, right, between the rural and the urban phenotype. And so this would be an instance of seeing the occurrence of evolution. And then these two mechanisms can, of course, interact. And so we could see the evolution of plasticity. So where the reaction norms themselves, so the slopes of what the lines you would draw between those points are actually slightly different. I'm sorry that keeps happening. I don't know what else to do. So I wanted to show you this framework first because I use it often in my research. And so in this case, I'll be showing you two different common garden experiments, and so I wanted to make sure that I covered this in detail. So when I'm referring to common gardens, that's what I mean. All of this stuff is gone too. But essentially, I'm going to talk to you about multiple different traits, and how they influence the persistence of this butterfly that is so good in comparison to other butterflies at persisting in these urban spaces. And so I have a short story on physiology, behavior, and morphology for you today. So for physiology, I am focusing on the thermal tolerance limits. So like other ectotherms, butterflies display a thermal performance curve where they increase in performance as temperature increases up until some optimum and then precipitously decline as enzymes denature and they become too hot. And so these thermal tolerance endpoints where performance equals 0 is what we're interested in today. These represent ecological death. And on the cold side, this cold tolerance value is our critical thermal minimum, or CT min. And then on the hot side, we have heat tolerance, critical thermal maximum, CT max. And these curves can evolve between populations, or they can even display plasticity through developmental acclimation. So what we might expect to see is that for urban populations, we might see that they have a shift in their curve towards hotter temperatures, so they can tolerate more heat, and theory often predicts that cold tolerance will get dragged along and that they will have a loss of cold tolerance. And in general, that's what we see when we look at urban versus rural populations in many ectotherms. So these are just some of the species in which folks have looked at thermal tolerance. But most of the time, we're not able to actually distinguish whether it's a plastic effect or an evolved effect, since these studies have not done the genetic work or the common garden rearing experiments in order to determine that it's from an evolutionary signal. So some studies have done that, including in Daphnia, but the majority of studies that have determined evolution are all in a single species of acorn ant. And so I just want to give a quick thank you to Osmarie, Jessica, and Dominic for their contributions to this work. So we were testing this question of whether heat tolerance has evolved or is plastic enough to help with urban warming in our cabbage white butterflies. So those are not cabbage whites, but they are painted lady butterflies, and they're in our thermal tolerance testing bath. And so we basically put them in these water temperature controlled baths, raise or lower the temperature by a degree per minute, and then see when they're able, when they stop flying. And so they get knocked out, kind of like this one in tube 2, and can't fly anymore. Well, you can see some of the ones in the back still fighting for their life as we're heating them up to near lethal temperatures. So, do we see evolution or plasticity in thermal tolerance of the cabbage white butterfly that can explain its high urban persistence? I will show you heat tolerance data on the top and cold tolerance data at the bottom. The rural populations are the lighter colored circles, and then urban populations are the darker colored squares. The two different rearing temperatures are on the x-axis, and then females on the left, males on the right. So we see that there is a difference between the rearing temperatures, so that they have higher heat tolerance when reared at 30 degrees, but we don't see any differences between the populations that were significant. And then the same pattern is repeated for cold tolerance. So we're seeing a loss of cold tolerance when they're reared at 30 degrees. They can only tolerate 5 degrees versus 3 degrees. And so this result suggests that there's no evolution going on in either heat or cold tolerance, since the populations are showing the same levels on both of those. Excuse me. But there is plasticity in the expected direction. So as you develop at warmer temperatures, you can tolerate more heat, and you're also losing cold tolerance. And while this degree of plasticity would undoubtedly be helpful in navigating warmer urban spaces, I don't think this explains why this butterfly is so good in comparison to other butterflies, because the plasticity was only moderate. So in rearing them at 10 degrees Celsius difference, we only saw a 1 degree Celsius change in heat tolerance and a 2 degree Celsius change in cold tolerance. So they can tolerate about a degree more of heat than when they're reared in warm temperatures. But urban heat island effects aren't, they're not necessarily experiencing 10 degrees Celsius difference between their oviposition sites. And to have 1 degree Celsius change for that, most urban heat islands are more on the magnitude of 3 to 4 degrees. So while it's helpful, it's not explaining the full effect of how they're able to deal with this urban heat. So that got me thinking about, well, what other mechanisms could they be using to cope with this urban heat? And I started to think more about behavior. And so specifically thermoregulatory behavior, because physiological evolution can be slowed down through behavioral thermoregulation in something known as the Bogart effect. And this has been classically studied in lizards, but it would apply to other ectotherms as well. So we can imagine our butterfly lizard hybrids, and the top panel is showing one that lives in a thermally homogeneous environment. So it could be like a thick jungle, right, or something where there's a lot of understory. And that butterfly lizard doesn't have a lot of options for picking between microclimates because of that. So what that does is when there are extreme temperature events like heat waves or cold snaps, it is exposed to those extremes, which leads to a faster pace of thermal tolerance evolution. On the bottom panel, you can imagine now this butterfly lizard lives in a heterogeneous environment. And so it has a lot more options for actually moving between sun and shade or cool and warm microclimates. So now when those cold snaps or heat waves happen, it can behaviorally regulate and buffer itself from experiencing those extreme temperatures. So now it can actually shield itself from selection, and it leads to a slower pace of thermal tolerance evolution. So this potentially makes physiological evolution unnecessary in order to cope with urban heat if they're really good at behaviorally regulating. So I was interested in this question. Oh, sorry, I'm moving forward too fast. I was interested in this because not only do I think it could explain the previous result where we didn't see any evolution of thermal tolerance and physiological thermal tolerance, but I also think it sets up an interesting question to see, is the behaviors, right, between urban and rural butterflies different across these urban gradients in terms of their thermal biology? And so I would expect this. I would expect that the two populations would overlap in their realized body temperatures. So as the environmental temperature changes, butterflies are still at the whim of their environment. They can't be perfect thermoregulators, so they kind of display this more thermal conformer classic curve. But we would expect to see that the body temperatures for the urban and rural overlap. And this is because the literature on thermal physiology shows that thermal preferences don't really differ between urban and rural organisms when folks have looked across different taxa. So I'm expecting them to want to be the same body temperatures and to be good enough at behaviorally thermoregulating to achieve those temperatures. But the underlying behaviors that they use to achieve this could differ between urban and rural individuals. So I want you to imagine for an example scenario that in a simplified thermal environment where we have a warm microclimate and a cool microclimate, and I'm showing a rural butterfly here as a darker gray and an urban butterfly as the brighter white. And that is to represent that by developing in a cooler rural environment, there could be a level of plasticity that causes the rural butterfly to have traits that increase its rate of heating or to achieve higher body temperatures. So we might see that in the same conditions, this butterfly would actually be heating up faster and would maybe move into a cooler microclimate to still achieve the same body temperature as the rural or as the urban butterfly. And I think there's going to be a lot of missing stuff, but okay. Yeah, these behavioral differences would be important for overall fitness by impacting potentially flight activity. So organisms have to balance, potentially the thermoregulatory need with flight. And if we reset our scenario, we could imagine that instead of having to move into the shade earlier at a hot point in the day, if this is early in the day when they're trying to heat up from, being resting overnight, a faster heating rate could actually allow that rural butterfly to reach a body temperature to sustain flight, and it can sooner take off into the environment, which will increase fitness by increasing the amount of time that it has to fly around to find mates or lay eggs. And so IB was interested in testing, are these behavioral differences underlying these body temperatures? Do they achieve the same body temperature? Let's find out. So I put butterflies in a simplified thermal environment outside in these enclosures. I gave them access to half of an enclosure of sun and half an enclosure of shade. And then I recorded the environmental conditions of light and temperature during these observational trials. I recorded the behavior in terms of their microhabitat choice. So are they sitting in the sun or are they moving into the shade? And I also kept track of their body temperatures in a non-invasive way by using thermal camera to take thermal photographs. And so I want to thank Tiffany, Vivian, and Augustine for their work on this project. So Tiffany was one of my capstone students that I mentored. She did this for her honors project. And then Vivian and Augustine were both high school students from the area as well. And so with them, we analyzed thousands of these thermal photographs that I'm showing you an example of there, in order to get an idea of, are the body temperatures the same and how do those link up to our underlying behaviors. Okay, so we did find that the body temperatures were similar. So I'm showing you females on the left and males on the right. The x-axis is the environmental temperature in the sunny side of our enclosure, and then the y-axis is the maximum body temperature that the butterflies were achieving. The different colored lines show the urban and rural populations, and they're overlapping with no differences at any given environmental temperature. And so that's all well and good, but what are the behaviors that they're doing underneath this in order to achieve those same body temperatures? And I was surprised to find that when we looked at microhabitat choice, there were no differences. So despite whatever traits that they had and developed from their respective environments, urban and rural butterflies were exactly doing the same things. They spent similar amounts of time in the sun and the shade in order to achieve those body temperatures. But what was most surprising was that that actually had an effect on activity. And that was shocking because I would think that there would be a thermal reason for this, but it doesn't seem like there is a thermal reason for this, and that urban butterflies just tended to fly more. So butterflies love to rest, which is super relatable, especially coming from Pacific time. I am just today waking up at a normal time, but the urban butterflies are resting less, and it doesn't relate to the differences in body temperature or the differences in the microclimates. So I wanted to dig into this a little more to see, like, when is that flight activity happening? Is it at specific body temperatures? And so I looked at, now we have body temperature on the x-axis, and then that proportion of time spent flying on the y-axis. And so you can see this effect a little bit stronger in males, but they're actually starting to fly at lower body temperatures. And so we're seeing that this increase in activity could be really interesting in a natural setting, and I'd love to follow up on this because it potentially in a natural setting, this could expand their activity window. And so, for example, if urban butterflies are losing activity in the hottest part of the day, well, they might be able to compensate for that because during the earliest parts of the day, they can potentially start flying sooner. So that was really fascinating to find. And like I said, I really would love to follow up on that result. Behavior, I'm sure many of you know, can be very complex to study. But this non-thermal likely driven pattern, whereas they're just kind of flying more and I can't necessarily explain why, got me thinking about my last little story about morphology. And so in this part, I'm going to be using common garden experiments again to test for morphological evolution of flight performance to see if they're just developing wings or body sizes that help them support that increased flight from our last study. And so here I'm looking at the butterfly's body size as well as the butterfly's wing sizes. And when we talk about size in an urban context, the general expectation is that ectotherms are going to get smaller. And so this is a study that looked at community sizes across a range of ectothermic taxa, and that's generally the pattern that we see, is that they tend to be shrinking or going into those negative numbers. And this could mean that individual species are getting smaller, or in this case, communities could be composed of smaller organisms themselves. And both have been shown in the literature. But this specific study also pointed out some outliers. And so in this case, a few taxa actually are getting larger when you look at urbanization. And so grasshoppers, macro moths, and butterflies all shifted to have larger body-sized communities. And the hypothesis that these authors put forward was that they believe, I'm sorry, it's cut off, they believe that the need for increased dispersal capabilities in a fragmented urban environment is going to outweigh the temperature effects on size. So when you're a smaller ectotherm, you have less metabolic costs, and in a hot city, that's beneficial, which is why we see things shrink. But for these taxa, dispersal is so important, right, to move around, that they actually are getting bigger in order to move between these fragmented habitat patches. So I want to see if this is what's happening with our Pyrus rapi cabbage white butterflies. And so when I talk about getting bigger in urban areas, I just want to make sure that we're not expecting the temperature size rule to go away. So the phenomenon where are higher temperatures, ectotherms tend to get smaller in size. So we're still expecting to see that plasticity where they're decreasing under the hotter common garden, but we might see something like this where there's an actual decrease in plasticity in the urban areas. So instead of getting very, very small at hot temperatures, they might just get slightly smaller. We could also see a consistent evolved response. So in this case, we could see that that temperature size rule holds for all of our populations. They're getting smaller to the same degree, but now our urban population is just overall bigger. So there's been an evolved response that's consistent across temperatures. And in this study, I was able to also include suburban. So you'll see these 3 colors in our results figures. And I want to shout out UNR undergraduate Brooke Fitch. I worked with her on this project for her honors thesis capstone as well. And so this would not have been possible without her. And when we're looking at the wing area, we found that there was that expected temperature size rule, that we had smaller wings at the higher rearing temperatures, but no real differences between the populations. The suburban, rural, and the urban all had the same degree of plasticity. So no evidence for evolution with wing size, at least, to support that they might be evolving larger wings to drive more flight. And the same pattern held between the forewing and also the hindwing of these butterflies. For our body mass result, this is where we found something kind of cool. So again, we see a decrease in body mass with temperature increasing, but in this case, we actually saw that there was a similar level of plasticity between the rural and the urban butterflies, but the suburban butterflies had a significantly higher level of plasticity. So that was kind of puzzling. And so what Brooke did that was really interesting and novel with this was she decided to look at the different body segments. So we split this mass out into head, thorax, and abdomen. And I'm going to talk about just the thorax and abdomen today and see where is this increase in plasticity for the suburban population coming from. And the answer is not the thorax. So we didn't see the difference in the thorax. This had a different kind of interesting thing where the males were actually more plastic than the females. So we see a larger decrease in the squares where the males are in comparison to the circles. So that could have some sex-specific effects for how the male butterflies are navigating the urban environment, since the thorax is where their wing muscles are. But where we saw the same plasticity difference was in the abdomen. So here our rural and urban butterflies are, sorry, my next slide's just showing completely black, and I know that's not right, so we'll get there. The urban and the rural butterflies showed very little plasticity, but the suburban butterflies showed a very high level of plasticity. And so this, I think, could potentially have implications for reproductive activity or something like that. And so I think it's a really cool thing to follow up on. And I don't have time to talk about other places, but in other experiments I've done looking at other traits, the suburban level has also shown kind of interesting things. So it doesn't seem like this trait is just a one-off difference, and there could potentially be realistic and real effects of maybe different combinations of stressors that coalesce at the suburban level to cause these types of patterns. So I definitely want to follow up on that in the future. Whoa, that's ugly. So how are cabbage white butterflies so successful? Well, I've told you a couple different stories, but I think what it comes down to, this is all going to be ugly, so I'm going to just put that up there and talk. I think what it comes down to is plasticity, right? This butterfly, there weren't any real strong evolved effects anywhere, but in both its physiology, right, it can tolerate slightly more heat when raised in a warm environment. When in terms of its behavior, they seem incredibly behaviorally flexible. They're doing the same kinds of behaviors and achieving the same body temperatures. And then in terms of the morphology, even though that really didn't show any strong signal for supporting the increase in flight that I found, it did show that the body mass and the wing size were plastic. So those butterflies would be developing smaller body sizes and potentially having that beneficial decrease in overall metabolism at hotter temperatures. So beyond the cabbage white butterfly, though, I just want to circle back to this thought about these multiple stressors. And, you know, a lot of times when we think about our stressors, we focus on just one of them at a time. And so I focused on urban heat today, but you can see where I think that it's important to integrate not only multiple stressors and think about, oh, this reaction to heat, it could also potentially affect their ability to deal with fragmentation. And this is also ugly, but multiple traits, I think, too, are going to be explaining these. So I think that in order to move forward, it's really important that we're looking at these stressors in combination and looking at populations in terms of multifaceted approaches that these species can be taking. And I'd love to continue doing this framework in additional species. And when I hopefully have my own lab, that's exactly what I will be doing. And I really thank you so much for your attention and for bearing with all of the ugly and cut off things. I want to thank all my collaborators at all of my different agencies, like the U.S. Fish and Wildlife, the U.S. Geological Survey, and the Xerces Society, as well as my university collaborators at the University of Nevada, Reno, and Case Western Reserve. And if there's time, I will absolutely take questions. And oh, you can also always feel free to email me. My email is up there. Thank you so much.
Okay, thank you, Angie. It's time for questions. Or for leaving.
Yes.
You mentioned lots of different things that could be impacting things, all of these factors. But one thing that you didn't mention was predators. Do these sound like something that, I don't know, songbirds or something would eat a lot of? It just struck me that the difference in how much time they fly would have a lot of, could be very sensitive to how much threat of predation there is.
Yeah, that's a good question. So I don't do as much community stuff. I'm more of a population ecologist. But you're right. From my knowledge, you'd be surprised. I think these, because of the compounds that they're eating as caterpillars, they don't actually get to tend to be eaten by a lot of birds. That's from the literature. That's not my own personal observations. So yeah, folks say that for how abundant this butterfly is, they tend not to be as predated upon as you might expect. But it's definitely a future area for research, especially in these urban spaces where there are a lot of birds, and I have folks who study that, exactly what you're talking about, like the predation of other caterpillars and the butterflies too. But it's a good future direction. I don't have any information about predators outside of that. Folks say they don't get eaten as often as you'd think.
Thank you. Yeah, thank you very much for your interesting talk. Well, my question is regarding the Boger effort and the evolution of the city max and city min.
Mm-hmm.
You mentioned all your designs are regarding or are concerning with city max, but what's about coal adaptation, the other side of the coin?
Yeah.
Because obviously is probably more labile trait, as you know, and also is not so constrained by Boger effect because yes, the microenvironment is not so different in minimum temperatures. So what's about that? Do you plan to make something?
No, you're absolutely right. So when I talked about the curve moving, I mentioned that it was oh, people will think that sometimes it just gets dragged along, but it absolutely could be doing its own thing. And so we didn't find any evolution of CT min either in the cold tolerance. We just found that it was plastic, and you're absolutely right. It displayed a greater level of lability, right? Like it was more plastic in the CT min trait. But yeah, in this species, there was no evolution of cold tolerance either. But I know that in other work that I've done from grad students at Case Western Reserve have studied like the cold tolerance and heat tolerance evolution in winter conditions, which is more understudied and is just starting to become a thing that folks are really starting to focus in on as well. Because yeah, it could potentially be more impactful in the winter. And so I can't remember now off the top of my head what the result was, but I believe that there was kind of an interesting facet where we didn't see the same evolution of heat tolerance in a species that heat tolerance had been found to evolve in summer conditions, but we found that the cold tolerance evolution was maintained between urban and rural.
Gracias, thanks.
Yeah.
Hi, thank you very much for the wonderful talk. And so I have a question because many of the results you've shown seem to have quite strong differences between males and females.
Mm-hmm.
So would this mean, when we talk about population, ecology, and vulnerabilities, that males are more or less vulnerable than females to some stressors, and this may change sex ratios, and potentially this may be per se vulnerability of not being able to reproduce, and more generally, what are the consequences of these strong differences between the two sexes?
Yeah, so the differences, thank you for pointing that out. I didn't have time to focus on a lot of the sex stuff that I did, but the differences are all expected based on the known dimorphisms between the species. And so there was nothing surprising that we found in that. Yes, and so in terms of how that would affect the vulnerability, I think absolutely it could. So like, for example, if like body size is filtering out, and so that's where we'd expect to see kind of maybe strong selection towards maybe the other direction, right? Where it's like we might see dimorphism actually decreasing if males are displaying a better, more suitable trait in a changing world. But I don't know about that. So most of my work is just looking right at the contemporary responses and hasn't really worked on forecasting forward, but I think it's a really important future direction. And one of the very first things I hope to do is to look at sexual selection in the city because it's often very understudied.
Thanks so much. It was really interesting, all the complementary experiments that you did, looking at physiology, behavior, and morphology. I was curious as the general framework that you use, I was a bit surprised to see how you confront plasticity to evolution, in a sense that they're not different mechanisms, and I wonder if it would be perhaps you could perhaps use the classical model of G by E, genotype times environment interaction, that it's a well-established system in the sense that plasticity itself evolves.
Yes.
So when you have shifts in reaction norms, you're seeing the evolution of plasticity. So
yes.
So I'm not sure it's really helpful for you to oppose plasticity and evolution, because they're not opposable concepts.
Yeah.
And whether it would be more useful to use a well-standardized, framework of genotype times environment interactions.
Yeah, I'm sorry if that was unclear. I was a little thrown off by all my slides looking different. That is what I'm doing. And I simplified like the plasticity and evolution. I couldn't show all of the possible reaction norm evolutions on like the slide. And I was thinking about it actually this morning. I was like, maybe I should change that in my talk. But I'm not seeing them as like these completely distinct and separate things. Like I normally mention like these obviously interact. Plasticity can affect the occurrence of evolution, right? So if something is plastic, it might actually forestall evolution. But also, yes, plasticity can evolve. And that was what one of the things I was trying to get at. So I am using a G by E interaction when I'm testing for these traits. I'm sorry if that was unclear.
No. It's just that, plasticity doesn't just happen. It's usually the result of natural selection. So it's by itself, an evolved trait. But
yes.
Yeah, thank you. So, do you have data on the genetic diversity of urban and rural populations? So, in other words, are you comparing different genetic pools or not?
Yeah, that's a good question. I haven't been able to do genetics. That was something I wanted to do, and to look at population differentiation across these gradients. I did not get the grants to do that when I was a graduate student. So I have samples in the freezer waiting to go. I would love to do that, because I think that gene flow would play a really important role in this system, since butterflies are fairly mobile. The best that I can tell you is that I have looked at like broad sense heritability by looking at the relationships between those siblings. And some of these traits are at least showing that they're heritable and like distinct in that way, but I don't know about the underlying genetics, and it's something I absolutely have been wanting to do for years. Thank you.
Okay, so if we don't have more questions, let's thank the speaker again.